Click for IB Biology. We're looking at the organelle known as the Golgi apparatus, vesicles, and some of the protein molecules involved in allowing those vesicles to form. There's a blank PDF of this presentation down in the description of the video below, where you'll also find 10 questions with answers and perhaps some other resources. So let's take a look at this Golgi apparatus. Golgi was a histologist who hypothesized the existence of this structure, although he personally never actually saw it. It was named in his honor. We begin the story. We need to go back 2 or 3 steps to appreciate the functioning of the Golgi apparatus. And let's begin with the nucleus here. We can see a micrograph of the nucleus there, and the production of messenger RNA. The gene is expressed, perhaps a gene for, let's take an example, a gene for the protein insulin. And so the gene is switched on and we make a copy of the gene called messenger RNA, and that will locate itself onto one of these ribosomes. So it leaves the nucleus through the pore, locates onto a ribosome, and the protein is built in that location. The protein then enters the endoplasmic reticulum tubular system and is moved around, and as it does so, it will perhaps be modified in some ways. And so we end up with the protein ending up at the end of the rough endoplasmic reticulum. And there a vesicle is formed by the budding off of the tubular rough endoplasmic reticulum to form a vesicle here, a membrane, single membrane fine structure. And this then moves across to the Golgi apparatus. So this has another layers of membranes, and they're shown more clearly in this image here, and then the micrograph is shown here. This stacking of membranes is indeed actually known as a Golgi stack. And variously, the size of those can vary between 20 nanometers and 200 nanometers. So notice that we're down to very small units of measurement again. So the vesicle with the protein in it, in our story, has moved towards the Golgi apparatus where it fuses, and the protein is now added into the Golgi apparatus, and it makes its way through the tubular network of the structure where it's modified. And typically what happens to proteins is that they often may have sugars added to them. So sugars are added onto the protein to give us a structure called a glycoprotein, or indeed they become phosphorylated with phosphates added to the protein is another classic modification that takes place in the Golgi apparatus. So the Golgi apparatus creates an isolated zone for the chemistry of forming glycoproteins and phosphorylation of proteins from ones which have been synthesized on the rough endoplasmic reticulum. And then the vesicles will then bud off, move towards the cell membrane where it fuses with the cell membrane and then releases the contents, the glycoprotein or the phosphorylated protein, into the external environment. In the case of insulin, this would happen to the lumen of the pancreas by a process which is called exocytosis. The vesicles that we've just been discussing, then there's 2 processes for us to think about. Exocytosis, exo, exit. This is where we're releasing substances out of the cell, and that's already been discussed in the previous discussion. We do have videos elsewhere about the process by which membranes can fuse. So if you look up the membrane part, cell membrane part of the course, you can actually find about how it is that membranes can fuse in this manner here to allow them to join together. Now, interestingly, if we come across to here, there's another process called endocytosis, which is the reverse, which is that we have a particle outside of the cell, and it's possible to get this into the cell. And the way that this can happen, in this particular case of endocytosis, is that the membrane will form a pouch like this and engulf the substances to be taken in. And these substances taken in are usually relatively small molecules in a kind of non-discriminatory manner from the environment, not to be confused with phagocytosis, which is a different process in white cells for engulfing bacterial pathogens and so forth. So this endocytosis is a way in which the cell can grab molecules from around it, and then it becomes enclosed within a vesicle in the process of doing so. Now, in order for this to happen, the cell membrane is here, and the membrane folds inwards under the influence of some molecules called Clathrins, like this. And the actual name of this protein molecule, which I've tried to draw here, is called a Clathrin triskelion. And they form around the membrane, the cell membrane, like this. It creates this pouch, and they form this supportive lattice work, like a matrix of these molecules, which allows the membrane to fold inward. So we have the lattice of the Clathrins. And then there's another molecule called Dynamin, which causes the entrance to become pinched tightly together. The cell membrane fuses, and so we have our vesicle formed internally with the contents in here, like so. Endocytosis using Clathrin triskelions. Very interesting. The links now are appearing to other videos, to the playlists, and of course to the channel should you wish to subscribe.